Hello students, this is Dr. M. Bhushanam, Assistant Professor in Zoology, Maharani Science College for Women, Bengaluru. Hope you're all doing well. Take care of your good health during this pandemic time, students. Well, in today's session, we are going to understand the concept of reproduction and development related to sea star, the type study animal under phylum Echinodermata. The objective of today's session is to understand the process of reproduction and the development or life cycle related to Asterias or sea star. The content includes reproduction and development pertaining to sea star. Well, students, starfish is an echinoderm which is an exclusively marine form. So, the process of development and completion of life cycle takes place exclusively in the marine water or in the aquatic medium. When we look at the organism of Asterias, it shows the condition of sex separation. Mean to say, the male and female gonads are present in two different organisms. So, males are different from that of the females. This condition we call it as sex separation. It is also called as dioecious or gonochoristic condition. All the terms indicate the gonads of two organisms of male and female are present in two different organisms of the sea star. Even though the sex separation is seen, both males and females does not have any difference of characters externally. So, when we look at the morphological details of an adult male or an adult female, there won't be any difference at all are seen. Then when this difference is seen, it is seen only during the time of breeding season of the organism, where the gonads will show the change in its color. So, that's about the importance of the sea star, adult male as well as female. So, when you look at the diagram down towards the left side, you can find how the ovary color is and how the testis color is. So, except for the color of the gonads, rest of all the features resemble same, including the body color. So, the sex separation, even though is seen, we cannot make out which is male and which is female. The second important feature is that the males and females of Asterias show a very primitive type of reproductive system in them. What does this term exactly refers to? It's a condition wherein the reproductive system itself will contain only the reproductive gonads. Rest of all the parts such as accessory organs like copulatory organs, receptacles, storing of the gametes like sperms and ova, all this doesn't present in the do not present in the body of the organism of Asterias. When very primitive kind of reproductive system and organism possess, it will not have the process of copulatory organs and the presence of copulatory organs and even the reservoirs for this ova and sperms to store. So such parts will be totally absent. Hence they are called primitive type. Then how are the gonadal structure is? These gonadal structures, when we look at, both the males and females will show presence of 5 pairs of gonads. If it is a male, it will have testis lobes as gonads. If it is a female, it will have ovaries as gonads. Both testes and ovaries are present located at the base of each arm. When you look at the structure there, here in the diagram down, towards the left side above, you have horn-like structures which are labeled as gonads there. So it could be a male or a female, whichever the gonad it be of, it resembles same. So it is present at the base, attached into the base of the arm. So like this, in each of the arm of 5, you find 5 pairs of gonads. So that's a specialty related to the gonadal structure of the organisms. Now, how are these gonads? Gonads are morphologically, when we look at, they appear to be like that of a bunch of grapes, or it could be like that of tuft of tubules. The size of these gonads will vary. They are very large at the time of breeding season. Otherwise, morphologically, testes and ovaries will look similar. They open into a common duct, which we call it as gonoduct, and this gonoduct will open by means of the opening called as gonopore, which extends towards the arboreal axis. When you find the axis of the body down as oral, the upper part as arboreal. So towards the arboreal axis is the gonopore, which gets extended, and through that opening of gonopore, the gonadal cells or the sex cells will come out. Testes are generally greyish in color and ovaries are pinkish to orangish in color, and this color difference we find only during the time of breeding season. It appears as a bunch of grapes of structures inside the base of each arm. The structure of gonads, gonoduct, gonopore together corresponds to the actual complete gonadal pair. So like this, we have 5 pairs of gonads present in each of the arm of the organism. In both the males and females, the location of the gonads, the actual gonads, is seen suspended inside the coelomic cavity of the arm. So that's the position related to the gonads of the organism. We understood that both the gonads will open or join into a common duct called gonoduct that will have an opening called gonopore through which the gonadal cells are thrown out of the body or released out of the body. This gonopore is present towards arboreal axis. So when you look at the body of the organism at arboreal axis, you find 5 openings of the gonopore through which the gonadal cells comes out. So that's the importance related to the gonads of the Asterias. Now, the process of reproduction will start at the season of breeding. So we call it as breeding season. The breeding season in case of Asterias is between February and April of each year. So the organisms of Asterias will reproduce once in an year. So we call it as annual episodic form of organism. Mean to say once in a year the organism will reproduce. So when is the time? It is between February and April period. During the time of reproduction, during the breeding time, the organism will show difference in the color of gonads, as what we have understood earlier. Generally, the testis appears grayish, ovaries appears pinkish or pinkish to orangish in color. During the process of breeding, the first step involves in the organism is the production of gametes. Otherwise, it is called gametogenesis, formation of gamete. If it is in males, we call it as spermatogenesis, where the production of sperms takes place. If it is in females, oogenesis is seen, where the production of ova or the eggs are seen. Sperm structure, when we look at, it is said to be dinoflagellate cell having an haploid nucleus in it. A cell which is globular or elliptical or oblong based on the stage of maturation, inside of which will be the presence of haploid nucleus of the set of chromosomes it will have. One of the end will show presence of 2 flagellae coming out. Hence, it is called dinoflagellate cell. The egg cell, when we look at the female gametic cell or the ova cell, it is also a haploid cell but contains the lecithin granules or yolk granules, the reserved food material granules, which are very less in content, less in quantity, but present throughout the cytoplasm of the egg cell called ooplasm. When you find lecithin granules evenly distributed in the cytoplasm or ooplasm of the egg, we call it as homolycytal condition. So we have sperms which are dinoflagellate. Over specialities, it is homolycytal egg. So such gametic cells are formed, are located inside the gonadal cells. Students, remember here the formation of ova or formation of the sperms, it takes place in the gonads. So, ova formation, sperm formation is seen in the gonads. But condition here is that, please underline, condition here is that production of sperm mother cells or ova mother cells, we call them as cells from which these gametic cells are formed, it does not take place inside the gonads. I repeat again, gonads will not contribute for the cells from which the gamete cells gets produced. So then where is this site? The site of production of this gametic mother cells are in the axial glands of water-vascular system. You remember the stone canal, which will have association with the axial gland? That is the place where the production of this gametic cells or the mother of these gametic cells are produced. So, sperm mother cells, ova mother cells are produced from the structure of axial gland. But formation of sperm, formation of ova from those mother cells or respective mother cells takes place in the gonads. So, gonads contribute for the site where the ova or the sperms are present. But their production, when we look at, their formation, when we look at, it is not contributed by the gonads, but it is by the axial glands associated with the water-vascular system. So, this important point you should remember. So, during the breeding season, Asterias, as we have seen, will breed once in a year between the month of February and April. And gametic cells come out of the gonads, then gonoduct, then through gonopore it will be released out. When we look at the mature gametic cells or sex cells, namely the sperms and the ova inside the gonads, the maturation of the gametic cells is purely decided by the nutrient that gets accumulated in which of the organs? You remember we discussed under the digestive system. It is in the storing cells related to pyloric caecae of digestive gland. Students, remember, in this pyloric caecae there are storing cells. So, amount of nutrients that get stored inside this storing cells of pyloric caecae or digestive gland, that in turn provides energy for the cells or gametic cells, is the deciding factor for the production of gametes and maturation of gametes. So, axial glands need to produce the mother cells which is under the control of the structures of digestive gland. I repeat again, the production of these gamete cells and the maturation of these gamete cells will take place under the control of the nutrient amount that is accumulated inside the digestive gland, that is pyloric caecae of the digestive gland. Say supposing if the nutrient content is less in an organism, it will not enter into the stage of production of gametes at all. Are you able to understand? So, if the nutrient quantity is more stored inside the body of organism, only such organism will enter into the stage of breeding, where the production of these gametes takes place. It is seen that after the production, so production is under the control of what? It is under the control of stored nutrient material in the body of organism. The second important factor is that release of the gametic cells. One is production. So we have understood production site itself is different. It is in the axial gland of water vascular system. But after the production, they come to, the mother cells will come to, gametic mother cells will come to the gonads. In the gonads, they form the sperm and the ova. After the production, which is under the control of nutrient content. Now, after the production, these structures of gametic cells need to come out of the body of the organism. So this is called release of gametic cells from the body of Asterias. Students, remember, the release of the sperms or the ova is under the control of the second important structure called as neurohormones of radial canals. You know, in each of the arm, there runs a canal related to water vascular system called radial canal. So that radial canal will control the release of gametes that are stored. Now, so there is a neurohormone secreted by this radial canal that will induce the process of release of the gametic cells. So it's a kind of stimulus that the organism should feel. Down in the picture towards the left side, you can see those white spots. That is the spot through which they are the gonopores through which the gametic cells are coming out. That white patches refers to the gametes that comes out of those gonopores. If it's a female, you find ova coming out. If it's a male, you find sperms coming out. It is seen that males produce innumerable number of sperms. It is estimated that per ejaculation around 250 millions of sperms are released by the male organism. If it is a female, it will release around 2.5 millions of eggs. So this we call it a spawn. Spawning means in groups, in bunches, in clusters, the eggs are released into the environment. Now, the gametic cells of male called sperms are 250 million, eggs of 2.5 million, they come into the water, they are shed into the water. Now, what happens here is that the ova and the sperms that are released in the seawater, which is under the control of what? Under the control of neurohormones of radial canal. Now, after the release of these sex cells in the water, there comes another step of the development, which we call it as fertilization. Fertilization. So when you look at the diagram down towards the left side, it refers to the slide showing the dinoflagellate sperms. Towards right side, when you look at the first cell is the zygotic cell, which enters into the mitotic cleavages or divisions to form multicellular nature for the embryo. Now, the process of fertilization refers to the fusion of male and female cells, gametic cells. We call it a syngamy. S-Y-N. Syn refers to fusion. Gamy refers to gametes. So, gametes gets fused here, which we call it as syngamy. Now, after the fusion of male and female gamete cells, the haploid cells, the first stage that we get immediately after fertilization is the zygote stage. So, zygote stage is a single-celled stage which is diploid in nature. When you look at here, the egg and sperm gets fused to form the zygote, which is a fertilized egg or diploid cell, single cell, we call it a zygote. Now, where does the process of fusion of male and female gametes are taking place, students? it is outside the body of the adults. It is in the water medium. So, this fusion of male and female gametes that takes place in the water outside the body of the parents, we call it as external fertilization. So, fertilization is external in case of Asterias. It forms a zygote. Now, the zygote is a single cell stage which is spherical in shape with 0.5 millimeters of diameter, containing very little amount of yolk, but it is evenly distributed. When you look at the life cycle of or embryogeny of Asterias, it is said to be indirect development because there is the involvement of 3 larval stages, namely Diplorula larva, the second is Bipinnaria larva, and the third one is Brachiolaria larva. So, in the life cycle of Asterias, we find 3 larval stages. The first one is Diplorula, the second one is Bipinnaria, the third one is Brachiolaria larva. So, to form that larval structures, immediately after the process of fertilization, the fertilized egg called zygote will now undergo the process of divisions, which are mitotic, which is after a period of rest. So, this divisions of mitotic divisions, what we find in a fertilized egg called zygote is called cleavage process. Down in the picture, when you look at towards the left side, the second picture refers to the divided cells referring to the cleavage process. Whereas the extreme right, when you look at the first diagram, is related to the gamete. The gamete cells are fused to form in the second diagram single diploid cell, it is labeled, indicating it is zygote. The third diagram, where 2 cell stage we find where the mitotic division is happening, which we call it as cleavage, leading to the multicellular nature down for the organism. Now, the next important feature here is that after a period of rest, the zygote will show mitotic divisions, which we call it as cleavage. So, after fertilization, the mitotic divisions that takes place inside the zygote, we call it as cleavage. The pattern of cleavage, when we look at, it is said to be holoblastic and equal. Holo means complete. So, division of mitotic cleavage furrow is complete and divides the single cell into 2 cells. And the daughter cells that are formed after the process of the cleavage are said to be similar in their size. So, hence it is called holoblastic and equal. The speciality of this cleavage is to bring in multicellular nature for this unicellular zygote. Are we able to follow? So, this is about the importance of cleavage related to the organism of Asterias. Now, after the process of holoblastic equal cleavage, we find multicellular nature for the developing embryo, and in differentiated form of this multicellular group of cells are called as morula stage. What you call it is morula stage. Now, after the stage of morula, where a group of cells that we find, there occurs the next stage called as blastula stage. The blastula stage is said to be celloblastula in case of Asterias. It is a stage where all this multicellular morula stage, the cells will start moving towards the peripheral region or surface region, enclosing a complete cavity at the center, as what you see in the diagram down towards the right side. The second row, last two diagrams from left side, from right side. So you find a multicellular nature called morula, but all the cells are pushed towards the surface, enclosing a central cavity. So that central cavity we call it as blastocele, surrounding which is a single layer of blastoderm cells which are ciliated. So ciliated nature of ball of cells enclosing the cavity of blastocele will make those cells, the embryo stage, to move freely in the water. So this structure we call it as coeloblastula, or simply called as it with the name blastula stage. So the central cavity called blastocele is filled with the fluid, and this is a stage where the organism starts moving freely in the water. It swims freely in the water. After a period of time of swimming, the stage starts resting, during which towards the posterior side, group of cells starts showing embolic invagination. So invagination refers to infolding of cells. So ball of cells starts infolding towards the one side. It is like this. So this infolding will lead to the opening called as blastopore. I repeat, single layered cells of blastoderm now start showing an infolding, which we call with an opening called as a blastopore. This infolding now starts moving towards the opposite end. Are you able to follow? And this infolding is called as the embolic invagination. Now, this invagination of cells that enters inside the blastocele, showing the opening of blastopore, now starts showing a new cavity inside. This new cavity, we call it as gastrocele or archenteron. So the upper cavity we call it as blastocele, and this infolded structure inside will have, this portion it will have the cavity called as the archenteron. This archenteronic cavity also starts moving inside. So the opening of this archenteron outside is through blastopore structure. Now, we find the speciality of this two-layered structure. We have an outer layer which is ciliated, inner layer that lines the archenteron doesn't have the cilia in it. So we find two layers. Outer layer we call it as ectoderm. Inner infolding layer that doesn't have cilia is called as endoderm. Students, remember, this endoderm now starts showing extension laterally. So sidewise, this invagination structure sidewise starts showing the extensions or pouch-like structures. At the same time, the ectoderm, the outer covering of cells that divides, will make certain of the cells to come out of the layer into the cavity of blastocele. So this new layer that gets formed from the separated cells of the ectoderm as well as the endoderm is called as mesoderm layer. So mesoderm layer is formed majorly from the ectoderm, and limited cells of endoderm also contribute for it. So they separate out of the layer. We call it as delamination process. So this delamination process will make the free movement of the blastoderm cells to enter into the cavity of blastocele, and they arrange there to form mesoderm. Students, now you find three layers inside the structure of the embryo. Outer ectoderm, which is ciliated. Inner mesoderm, which is non-ciliated. and inner to mesoderm is the endoderm. So all these three germ layers with the cavity of archenteron or gastrocele is now called with the name gastrula stage. So down in the picture, the gastrula stage is very clearly represented. Okay. Now, the opening of blastopore starts showing an elongation that becomes the anus in the later stage of larva. And on the surface at lateral position there occurs one more opening of the body, which we call it as mouth. So when you look at the stage of organism at gastrula, the blastopore is forming the anus, and somewhere in between the lateral side of the body you find one more opening called the mouth. Between the mouth and the anus there arises the accumulation of this endoderm to form the structure of digestive tract, which we call it as stomodium or mouth. Along with the anus there forms the digestive tract, which is tube-like. Now, at the same time, the cilia which is present on the ectoderm, outer surface of the ectoderm of gastrula stage, now start showing the degeneration. So, the cells of ectoderm that had the attachment of cilia now is lost because a new cilia arranged layer gets formed, a running layer of cilia attached. We call it as a ciliary band. This ciliary band is formed from the structure of ectoderm itself, whereas the mesoderm gets formed from two layers which are there. It is both the endoderm as well as the outer ectoderm, but predominantly ectoderm will contribute for it. And this stage of development is called late gastrula stage. In late gastrula, we find endoderm showing the presence of mouth towards one side, anus towards the other side, with the endomic internal endodermic pouches, coelomic pouches, and the outer layer of the ectoderm, which is ciliated, and mesoderm layers or loose connective tissue which is dispended within the blastocele. So that's about the importance of late gastrula stage. Students, remember here the structure of digestive system is getting formed from the endoderm that gives rise to the mouth, the stomach, the intestine, the anus, etc. At the same time, endoderm also forms special kind of pouches within the body, which we call it as coelomic pouches. Students, remember, they are responsible for the formation of hydroenterocele in the body of the larva. Remember, this hydroenterocele is the structure in adult. It forms the water-vascular system. You know, water-vascular system is a modified coelomic cavity. So, which is the modified part that it forms? It is this hydroenterocoelom part. Okay, that's the importance of gastrulation stage. Now, soon after the late gastrula stage, there occurs the first larval stage, which we call it as Diplorula larva. This is also the first free swimming larval stage. Gastrula, after entering into the early gastrula and late gastrula stages, it forms the larvae because indirect development is seen. There are three types of larvae developed, three stages of larvae developed. The first one is Diplorula, second one is Bipinnaria larva, third one is Brachiolaria larva. So soon after the formation of late gastrula, the first larval stage formed is called Diplorula larva, which is also called as early Bipinnaria larva. Students, remember, this is a common larval stage seen in the life cycle of all the echinoderms. So the stage of larva which is commonly shared by all the echinoderms in their life cycle is this Diplorula larva or early Bipinnaria larva. The speciality of this particular larva is it is a free swimming larva, starts floating freely onto the water of the sea. So we call it as pelagic. It is transparent. The body is transparent. Meaning to say the internal content of the body can be easily seen from outside. The shape of it is ovoidal or elliptical, and they don't have any arms or arm-like structures connected to the body. It is globe-like, spherical structure generally. When we look at the larval stage of it, it is microscopic, free swimming. It shows presence of two major parts. Part one is the digestive system. The digestive system of this larva is complete and simple. Complete because it starts with mouth, ends with the anus. It is simple because it has very less number of parts. It is muscular, made up of muscles. The second important feature is that the presence of ciliary bands. Students, remember, when we look at the late gastrula stage of the organism, the outer ectoderm had the presence of cilia. That is the only layer where the cilia were present. But what happens now is that those cilia structures gets formed as a band. So band of cilia gets formed. We call it a ciliary band. So they are formed from which layer? Outer ectoderm. They are long chain of cilia in the form of bands that surrounds the transparent oval body. And it shows presence of perioral band. You can see that structure represented above the mouth, perioral band. And based on the position where this band is, we give the name for that band. So from which layer this particular structure of perioral band is formed? It is from the ectoderm of the gastrula stage. So this perioral band, nothing but a common ciliary band, helps the stage of larva to freely swim and float in the water. So very purpose of the oral band is locomotion. The second important feature of the ciliary band is to help the animal to have the feeding process. So when you look at the ciliary band at post-anal region, means to say below the mouth, you have the other ciliary band. So one ciliary band is present surrounding the region. Above the mouth we call it as perioral band, and below the mouth there is one more band which we call it as a post-anal ciliary band. The importance of post-anal ciliary band is to help the animal to have the process of locomotion, whereas perioral ciliary band helps, it is nearer to the mouth region, helps in the process of feeding. Please correct, students. Perioral band that surrounds the mouth area will help the animal to have feeding process, whereas pre-anal ciliary band, which is seen down, That helps in the locomotion of the organism. So that's about the importance of first stage of larva called Diplorula larva. It is the first swimming larva in the life cycle of Asterias. Not only Asterias, this is a common larval stage that we find in all echinoderms. The second stage of development, if Asterias, when we look at, it is called Bipinnaria larva. We have understood that the Diplorula larva gets transformed into the stage of Bipinnaria larva. What are all the changes that it shows? It shows changes with the presence of ciliary band and extensions of the body in the form of arms. The globe-like body, if I pull out, if I apply pressure and pull out, it forms the extensions which are called as arms. Generally, the shape of Bipinnaria is said to be ovoidal in shape only, but it will have the presence of arms attached. It has a transparent body. It is also microscopic organism, starts pelagic organism, starts feeding on the microscopic planktons. At the same time, the body of the organism shows two important structures. Number one, digestive tract. So digestive tract is complete. Excuse me. Starting from mouth, ends with the anus. It is simple. And the body is covered over by the presence of arms, which are nothing but the extensions of the body surface. There are two median arms which are unpaired. Median means at middle. Towards anterior side, top, you have one arm moving towards the dorsal side, one arm moving towards ventral side. It is like this. One towards the dorsal, one towards the ventral. So these two arms are called median arms. They are unpaired arms. Dorsal and ventral arms. It is represented with the name 1 and 8. Dorsal median arm, ventral median arm. And in addition to this unpaired, there are 5 paired arms. They appear in pairs. Such 5 pairs of arms are seen. Number 1, pre-oral arms present near the mouth. Post-oral arm present opposite to the down towards the posterior side of the mouth. So pre-oral arm, post-oral arm. The third one is posterior dorsal arms. Posterior dorsal arms. Next comes posterior lateral arms. The fifth one is dorsolateral arms. So we find 5 different types of paired arms based on the position. Let me tell you, or let me ease out the concept here. When I say dorsomedian, at the middle, median refers to middle, dorsal extension. So when this is the body of the organism of Diplorula, I'm going to extend it towards, I'm going to pull it towards the upper side. So towards dorsal side, if I pull one arm, it gets formed, and that is called dorsal median arm. Towards ventral side, if I'm pulling it out, it forms the ventromedian arm. Like this, if I pull out near the oral region, it forms a pre-oral arms. And towards posterior to the mouth, if I pull out, I call it as post-oral arms. The third pair is posterior-dorsal. Posterior refers to down, dorsal refers to back. So I'm going to pull them back. I call it as posterior-dorsal. Then posterior-lateral down. Towards the lateral side, I'm going to pull it. We call it as posterior-lateral. Then dorsolateral. So we call it as dorsal side, but laterally I'm going to pull it. So like this, we have 5 pairs of arms and 2 unpaired arms are associated with the body structure of Bipinnaria. Students, remember here, each of the arm is in association with the ciliary bands. So ciliated arms it is called otherwise. So this ciliated arms present attached to the body will help this animal to move freely in the water. So it is also an active stage of the larva of Asterias. So Diplorula larva is not only active stage commonly seen for all echinoderm, but of Bipinnaria when we look at it is the larva pertaining to the Asterias. It is the second larval stage. How it is? It is microscopic, free swimming larval stage, which is which will feed on the planktons. So down is the picture towards the right side, sorry, left side. Excuse me. It's the microscopic view of that Bipinnaria larva. Now, another stage into which the Bipinnaria larva enters during the development of developmental stage or developmental life cycle stage of Asterias when we look at excuse me, it is called Brachiolaria larva. This is the third stage of the larva in the life cycle of Asterias. So after 5 to 7 weeks of time, this Brachiolaria larva gets formed from or transformed from the Bipinnaria. So during the metamorphosis of Bipinnaria to Brachiolaria, the larva shows some significant changes in the body. It is initially free swimming. Larva is bilaterally symmetrical, and after a period of time, the larva of Bipinnaria enters into the stage of Brachiolaria. So Brachiolaria larva is also microscopic. It is also free swimming. It is also pelagic. It is also bilaterally symmetrical. Remember students, all the larvae of echinoderms are bilaterally symmetrical, whereas all the adults are